Good evening, everyone. I hope everyone is good. Everyone is fine. So we are back with a very important chapter, that is biomolecules. Now, in this chapter, why do I say this is important? The reason is that the number of questions that comes from this is real good. And one more thing is there, that because you do not have to do a lot of mehnat here, but still you get good number of questions. You can say 1 to 2 questions every year. It is fixed from here. So that's why this becomes very important chapter. So yeah, so now we are going to start with the next with the topic, that is carbohydrates. So what are the things that we are going to complete here? Carbohydrates we'll try to complete, and we'll try to complete all the important types of question. Then amino acids, and amino acids, after that there will be nucleic acid, then there will be proteins will be there, and the last one, that is vitamins and hormones. That is something that you already study all these parts in botany, zoology also. So I will not go much in depth, but still I will try to take all the chemical aspects of these topics, that is carbohydrates and amino acids. And at last we'll be just talking about few things, like if there is, because it will be my last session, so we'll be talking about few things. Chalo, so let's start. So what is carbohydrates? Carbohydrates is nothing but it is hydrate of carbon, as the name itself suggests. So what is the general formula? General formula, you can say that CxH2O whole Y. So some moles of X and few moles of Y. So in such case, what we can say, that this is the carbohydrates general formula. Like we have many examples. We can say that glucose, fructose, sucrose, these things are there. If you talk about glucose, so it is C6H12O6. So you can say C6H2O whole 6. And if you talk about sucrose, that is C12H22O11, or maltose, so this is going to be C12H2O whole 11. So these are the points that you should keep in mind. Very important. Next, we are going to study about classification of carbohydrates. If you talk about classification of carbohydrates, there is going to be sugar and non-sugar. What are the properties of sugar? Sweet in taste, crystalline and soluble in vital water. And there are how many types? There is monosaccharides, oligosaccharides. In this, there may be. Now see, if you are talking about monosaccharides, this may be aldose or ketose. Aldose and ketose, talking about the functional group. If there is aldehyde as the functional group, it will be aldose. Ketone as the functional group, it will be ketose. If you talk about oligosaccharides, oligosaccharide means telling that how many number of carbohydrate, or you can say these monosaccharides units are there in this. So if there are 2 units, there will be disaccharide. If there are 3, trisaccharide, tetra, you understand it. Up to 10 it will be called oligosaccharide, and after that more than 10 it will be called polysaccharide. Let's talk about non-sugars. Now they are generally also known as the polysaccharides because they are non-sugars. What are the properties? Tasteless, amorphous solids, and non-crystalline, insoluble in water, and these yield many sweet monosaccharide on acidic hydrolysis. If you do the hydrolysis, you will get monosaccharides. Now, classification on the basis of number of hydrolyzed products. I hope you all can see that if there is monosaccharide, so this will not be further, it will not be further hydrolyzed, or it will be giving just one or single unit. So you can directly say cannot be hydrolyzed. So monosaccharides cannot be hydrolyzed. Examples are glucose, fructose, galactose. Many things are there. Next, oligosaccharides, 2 to 10 units are there, like sucrose, maltose, lactose. If you talk about polysaccharides, there will be more than 10, like many units are there, starch, cellulose, gums, resins, etc. Now, आगे बढ़ते हैं. Now, classification on the basis of functional group, as I have already told you, it may be ketose or it may be aldose. If you are talking about aldose, it will be like that. कैसा होगा? सोचो. ये आपको ध्यान रखना है. So if there is, let's say there is aldehyde, so can you see that there is aldehyde, CHO group is there, and you can see CHOH, 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 CHOH. Let's say it is whatever is the number of CHOH, at last it will be CH2OH. If you talk about ketose, of course we know that for the ketose, for in both the side there should be some alkyl part should be there. So CH2OH, in between there is CO, there is keto part, and then there will be CHOH whole N and CH2OH. So this is what ketose. This is how the ketose will be looking like, and this is what aldose will be looking like. Examples, the best examples that we have are glucose for aldohexose, while ketose for fructose. Okay, that is aldoketose. We will see these things. Now, if you talk about carbon atoms, now classification of monosaccharides on the basis of carbon atoms in hydrolyzed products. Now, like carbon atoms, number of carbon atoms are here given. Now, general terms, and then aldehyde and ketone. Ketone will be there. Then what will be the name basically? Let's say there are 3 units are there. So what will be called? Aldotriose. For ketones, it will be aldoketose. Ketotriose, sorry. And if there is tetra, or let's say 4, so there are how many units? 4 units are there. It will be aldotetrose, that will be ketotetrose. Here, I hope you understand, so and so forth. Not a big deal. You all can see this. Okay. Now, we are going to see open chain structure of the monosaccharides. These are few important monosaccharides that we are going to study here. Now, and all these are going to be aldoses. Now, see, like D-glucose. In this, can you see that you all, I hope you know about it, how to give the capital D, capital L configuration, DL configuration, DL configuration. Have you any idea how to give that DL configuration? There are few rules, whatever I'm telling you, please keep in mind. There will be more than, consider, always remember, more than one chiral carbon should be there. The topmost carbon should be highly oxidized functional groups, or what we can say, either this should be CHO or it should be COOH. Okay, and at downside this may be anything like, you know, CH2OH, CH2OH, consider karo, CH2OH is there, consider. So consider it as CH2OH. Now you have to just see that whichever is the lowest chiral carbon. Which is the lowest chiral carbon? Here it is also lowest chiral carbon. Now इस पे क्या क्या चीजें होंगी? H and OH consider करो, and OH and H. If OH is at the right side, it will be called D. If OH is at the left side, it will be called L. That is how you give the DL configuration. I hope you all can understand it. But there is one more rule, though it is not relevant here, but still you should know. If there is amine, then you will not see that where is OH. You will only see the amine group. Amine is given higher priority. Consider that there is amine also. Amine, consider it as it is at the left side. So rather than telling D, you will call it L. I hope it is all making sense. But again, I'm telling you, because amine is not going to be used here, so we are not going to talk about it. So I hope it is making sense. So if OH is at the right side, it will be D. If it is at left side, it will be L. Okay. Now, D-glucose, why it is called D-glucose? Because I hope you all can see that there is OH, the lowest chiral carbon, that is here. That is what? That is here OH is what? At the right side. I hope. And what is the functional group here? Aldehyde. But is there any carbon where OH is at the left side? Yes, I hope you all can see that it is carbon number 1, it is carbon number 2. At carbon number 2, and it is what? Carbon number 3. So at third carbon, it is what? OH is at the left side. So what we can say, it is also called typical nature. They sometimes ask that in the glucose, which carbon has the typical nature of L? So we will say that third carbon is of the L nature. Though overall compound is D only. The reason being is that the lowest carbon, or you can say lowest chiral carbon, has the OH at the right side. Is that okay? Next. So if you talk about allos, so there will be few changes. Can you see OH? All of the OH are at the right side. So what you can say? There is no L, and all have same number of carbon, that is 6. If you talk about mannose is also having the second carbon also at L, and third carbon is also at L. So what we can say? First, second, third. So second and third, both of them are L. So what we can say? 2 and 3, the typical nature is 2, 3L. What about this galactose? Please, you can see that, and you can keep keep these things in your head. Now, what about the ketose? Ketose, we have all studied by now, is all aldose. Now we are going to study about ketose. Can you see here that it is fructose? CH2OH, CO, CHOH, CHOH. Can you see? Now, what we have, main things you have to keep in mind. This is not the chiral carbon. This is not the chiral carbon. How many chiral carbons are there? 1, 2, 3. 3 chiral carbons are there. Okay. Now, here, if there are 3 chiral carbons, which is something that we can say at the L side. Can you see? Third carbon is at the L. So what we can say? 3L is the typical nature is 3L. So I hope you all can understand it, how it is all working. Chalo, but all of these were what? Open chain. These all were the open chain structures. I have not told you still the cyclic, or you can say closed chain structure. There are closed chain structure also, and that is very important. Keep in mind. Next, we are going to study about epimers. What are epimers? Epimers are those, those kind of, these are optical isomers. which have at only one of the carbon, the configuration is different. Can you see? And you can see here that, can you see, this is what, this is glucose and this is what, mannose. Is there any difference? Can you see? CHO same. On the second carbon, can you see it is C2, it is called C2, and here it is C2. At C2, what is the difference? OH is here at the right side, here it is at the left side. All other are same. Can you just match it? All other are same. If all others are same, so in such case we can directly say that it is which kind of epimer? It is called C2 epimer. It will be called C2 epimer. If you compare this glucose with allose, what you will see that OH, this OH is at the right side or left side? It is at right side. So in such case, what we can say? So therefore the difference is at the third carbon, C1, C2, C3. So it will be C3 epimer. I hope you can understand it. Whatever is the difference, because here the carbon third is the difference. So therefore C3 epimer. So similarly, you can just keep on going and you can see that here it is galactose is C4 epimer. आगे बढ़ेंगे and we'll see reducing and non-reducing properties of the sugar. Let's say, what are the reducing sugars? If reduces Tollens reagent, Fehling solution and Benedict solution, it will be called reducing sugars. But if does not, so don't reduce Tollens, Fehlings and Benedict. So in such case, it will be called non-reducing sugars. Now, should have at least one hemiacetal or hemiketal functional group, all mono and oligosaccharides except sucrose. So यहाँ पर क्या होना चाहिए? At least one hemiketal, one hemiacetal or hemiketal functional group should be available. I will show you how this whole is formed, and then you will understand it how. What is this? Now, should have acetal linkage, etc. Example, all polysaccharides and only few oligosaccharides, which you can see sucrose is a non-reducing sugar. The reason being कि उसका जो, you can see the whatever is that what to say, that hemiacetal or hemiketal वाला जो functional group है, वो that carbon is already bonded. If that is bonded, that is C1, C5. You will see that because it is already bonded. So in such case, what we can say, that we can see that it is not going to be reducing sugar. चलो आगे बढ़ेंगे सीधा. Hydrolysis of sucrose. If you hydrolyze sucrose, it will give you fructose and glucose. But which fructose and which glucose? We have to remember this. I will show you what is that. Basically, it will give you alpha D or beta D. पहले आप ये सोचो. Alpha D glucose, but positive or negative? पहले आप ये सोचो. Alpha D positive. Alpha D, because glucose is positive, it is not negative. And alpha D, no, not alpha. Basically beta D. So alpha D glucose and beta D negative fructose. So alpha D glucose and beta D fructose. When we hydrolyze it, we get that. Okay. What is that alpha? What is that beta? We'll talk about it. It tells about the anomeric forms. These are what? Anomeric forms. What are anomeric forms? We'll talk about it. This is also a kind of what? Isomerism. Now D is something that is telling you about the where is that lower OH is there. Is it in the right side or in the left side? This positive and negative telling you about this positive is telling that it is what? It is dextrorotatory. For, and it is what? It is telling you about the levorotatory. It is levorotatory. So that's why it is negative. Here it is glucose, here it is fructose. Okay, चलो आगे बढ़ेंगे. Next, fructose is an example of pyranose, ketohexose, aldohexose, heptose. I hope you all know that this is what? It is ketohexose. आगे बढ़ेंगे. When the following aldohexose exists in its D configuration, the total number of stereoisomers in its pyranose form. Okay, first of all, can you see that it is ketose or aldose? It is aldose. अब देखो aldose का pyranose form कैसे बनाया जाता है? How do you make that? First of all, you have to understand it. Furanose and pyranose, two forms will be there. Furanose is less number, that is 5 carbon change, while pyranose is what? Is 6 carbon change. How do you make that? Just try to see that how many number of carbon are there? 1, 2, 3, 4, 5 and 6. And always remember, because we have 6-membered carbon is more stable. In such case, there is going to be a reaction of alcohol and aldehyde part. So basically, can we not think it something like that? Dekho, CHO. Just think about it. I'm going to do one intramolecular reaction. Just keep on seeing it. OH, CH, OH, and C, OH, H, CH2, OH. Now, can we see here? This is what? This is OH part. OH, and here we have CO, oxygen, delta negative, here carbon, delta positive. This oxygen, can you see that this is oxygen is having, this is what? Lone pair. So this lone pair can attack at the positive parts. What will happen as it attacks on the positive part? This will be, I hope we have studied that. Once OH, alcohol, reacts with aldehyde or ketone, it can converts into acetal or hemiacetal or hemiketal. I hope you all remember. So यहाँ से जब ये attack करेगा, what will be it looking like? It will be something looking like that. And next will be कि यहाँ से ये जो H plus वहाँ पे it will be forming OH. So how does this look like? Either C will be like OH, H, C, H, H, C, H, OH, C, H, OH, C, and this will be O, H, CH2, OH. Can you see that? So ये एक type हो गया. This is what, can you see here? How do you make the, how do you make this? What you have to do here? How beta and alpha is given. Here बीटा और अल्फा से बात छोड़ो, यहाँ पर सिंपल सी एक बात समझो कि यहाँ पर जो, can you see that how many of this are, we can say this is what, because it is telling that pyranose form. This is what pyranose form. This is what pyranose form. Can you find out how many of them are chiral carbon? Now this carbon which was non-chiral or achiral, can you see now after once it has taken its pyranose form, now this carbon has become chiral. Can you see? Because all the groups that is attached it is all the different. So what we can say, this is also chiral, this is non-chiral, this is chiral, this is chiral, and this is also what? Chiral. So how many of the chiral carbons are there? There are 4 chiral carbons. There are 4 chiral carbon. And is there any symmetry? No. It is what? Asymmetric. If it is asymmetric, so what we can say? In such case, we can say that number of isomers will be how much? Number of isomers will be equal to 2 to the power 4. That is, sorry, 2 to the power, how many of them are there? 1, 2, 3, 4. 4 are there. So directly we can say that, what we can say? There is 2, 2, 2, 2. So from here, because DL, 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 so we can say that there is going to be directly you can say this, it is going to be 8. Acha, what other things you have to think about it, and you have to keep in mind. देखो जरा यहाँ पे जरा सोचना कि how do you make that, or how do you make that in the pyranose form? How does it look like? First of all, try to make a 6-membered carbon chain. Can you see? चलो अब यहाँ पर जरा ध्यान से समझना कि what we can see. हाँ, so in its D configuration, the total number, okay, in its D configuration. Now, acha, it is asking for in its D configuration. So once it is in D configuration, okay. So it is only asking for that. So how many of the carbons we have got? That is, leave that one. Basically, we have to see 2 to the power 3. In such case, we can say that 4 chiral carbons are there. But one more thing we have to keep in mind is at 1, 2, 3 carbons are there which are chiral. If you think about D, means if OH is at the left side, okay, 2 to the power 3, that will be 8. Ultimately it should be coming, answer should be 8 only. Yeah. So now what we are going to talk about it here, the total number of stereoisomers. How do you this is something that we have done, 8. Now we have to see that how do you make that? What is the pyranose form? Pyranose form mein you just make the groups. Whichever is at the right side, that will be kept at the, down. If whichever is at the left side, it will be kept at the, upside. Because you can see that first chiral carbon has hydrogen up, OH down. As you come down here, H, it will be, so it is non-chiral. What about next carbon? It can be either OH may be up or down. Consider as agar OH, if it is at the right side, then OH will go down. If it is at the left side, it will be up, right? You understand it. If OH is downside, left side, it will be up, and because H is right, so it will be down. But as we come to this carbon, we have to always think about key, because now there is no left and right. CH2OH is at the left and H is at the right. Now see, it is at the down and it is, so you cannot say it is left or right. So directly think, in such case, whichever is more having more molar mass, CH2OH is having more molar mass, so it will go up. So CH2OH, and hydrogen having less molar mass, so it will go down as a rule. Now, अब यहाँ पर जब OH is at the left side, then it will be, or OH is at the right side, or you can say once the OH is right, it will be downside. It will be called alpha. But वही अगर OH अगर up आ गया, if the OH comes up, if OH comes up, if OH comes up, H comes down, everything is same. Rest everything is same. It will be called beta. It is called anomers of each other. Both of them are anomers of each other. And this is called the carbon that is, you can say, anomeric carbon. This is called anomeric carbon. Anomeric carbon. We also call it what? Hemiacetal or acetal carbon. Okay, chalo aage badhenge. Now the structure of D-glucose. D-glucose is this. Now the structure of L-glucose. How do you say D or L? I hope you all understand it. If OH is at the right side, the lowest OH is at the right side, it will be D. If it becomes left, it will be called L. अब देखो, ऐसा तो तुम कर नहीं सकते हो केवल इसको चेंज कर दो, then तो it will not be a glucose. You will have to convert completely the whole molecule into left. तो चलो, ऐसे में if everything right comes to left, then it will be called L-glucose. चलो जरा सोचो, can you see any of these example where all the OH and H gets inverted? Yeah, I can see that option A is here the correct option. आगे बढ़ो. Now chiral carbon atom in glucose and fructose are. Glucose and fructose are. चलो जरा ध्यान से देखो. In such case, in glucose and fructose. ग्लूकोज को देखो कैसा दिखता है, CHO, CHOH, 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 CH2OH. Can you see how many of the chiral carbons are there? 1, 2, 3, 4. So 4 chiral carbons are there in glucose. In glucose there are 4 chiral carbon. What about fructose? It is what? CH2OH, then C double bond O, C OH, H, C. Basically you have to just make this. Anyways, how many carbon? 3 OH, H, C, H2OH. What about that? Can you see how many of the chiral carbons are there? 1, 2, can you see? 3. Now this is non-chiral, this is also non-chiral. So how many of them are there? This is fructose, and fructose having how many? 3 chiral carbon. And there it is how much? 4 chiral carbon. तो अब सोचो, 4 in glucose and 3 in fructose. So option C is the correct option. चलो आगे बढ़ेंगे. Now anomers. What about anomers? Anomers I have already told you anyways. Anomers are diastereomers that differ in configuration at the acetal or hemiacetal carbon, that is also called anomeric carbon atom of a sugar, and its cyclic form, that is anomeric carbon, a carbon bonded with the 2 oxygen atom. For example, alpha D glucose and beta D glucose are anomers, and alpha D and beta D fructose are anomers. Like मैं glucose की बात करूँ, I am giving you example of glucose. Later on you can just think in the term of fructose also. How can we say? It is nothing but COH H. I am making the hemiacetal form, so do not think what I am doing कि ऐसा कैसे हो गया ये CHOH. Now here we have O, and then we have CH2OH, and here we have H. How many carbon? 1, 2, 3, 4, 5, 6. Okay. Now can we just try to make that? See this. Two things are possible. One is this. Another is going to be what? Can you see? Another is C. Same thing. OH, H, OH, H, H, OH, H, O. This OH just became at that side, which was at the right. Now it has come to the left. Now it is what? CH2OH. It is because OH is at the right side, you will call it alpha. It is capital D minus or positive plus glucose. What about this? OH is at the left side, beta plus or minus. Of course it is going to be plus. Glucose का ये तो है नहीं, बदल गया. Beta positive D or beta D positive glucose. And which kind of glucose? Will you write only glucose? No. glucopyranose, glucopyranose, because it looks like pyran, that is 6-membered, or glucopyranose. So that is how you call it. चलो जरा इसे देखो कि दिखता कैसा है. How do you call it, or how do you say what something is alpha or something is beta? जरा ध्यान से सुनना. Now just consider it. देख रहे हो? चलो अब जरा सोचो. The first carbon we have here is OH is at the left side or right side? Left side. Once left side, what I told you as a rule, OH should be at the up. Left should be up and right should be down. So H will be coming down. What about H and OH? Here at the next carbon is, of course, we can see that this is what? This is first carbon. Yes, is there? CH2OH. Yeah. So here next one is, can you see, this is hemiacetal oxygen is there. So this is going here, hemiacetal oxygen. See, now this is coming to the next carbon, that is C2. Think about this, C2OH. OH is at up or down? Can you see? Right. So right will be coming at the lower. So here OH is up or left, so it will be up. OH is right, so it will be down. Next, here it is H. Here it is already, it is attached to this carbon. Can you see next carbon? And what about this? This is attached to two things. One is H and one is CH2OH. Now I told you whenever this is indecisive, because it is not left or right, because CH2OH is down, it is not left or right. So in such case you have to see molecular mass. So whichever is having higher molecular mass, it will go up while rest will be coming down. So this is how you can just form. This is beta D glucopyranose. Glucopyranose. But what about alpha? For alpha, just you have to do what? If you just exchange this H to OH, so what will happen? This will become alpha D glucopyranose. I hope it is all making sense. That is how you have to make that. Okay, चलो आगे बढ़ेंगे. Next, monosaccharides, that is glucose and fructose. Now we are going to study specifically those points from where the question comes. We cannot go in detail to everything, but still we'll try, and then we'll go for the question solving. चलो आगे बढ़ो. Like glucose is the most common monosaccharide, is known as the dextrose because it occurs in nature principally as the optically active dextrorotatory isomers. No problem. Next, here we can see that beta form of D-glucose is more stable than alpha D-glucose. This is something that we can keep in mind. Next, आगे बढ़ो. Now, ring structure of glucose is due to formation of hemiacetal and ring formation between. Now, can we see ring formation में कौन-कौन से carbon आपके involve हुए थे? This is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So which carbon? Carbon 1, and which carbon? Carbon 5. Carbon 1 and 5 are attached. So therefore what we can say? C1 and C5, option A. Okay, next आगे बढ़ो. That alpha D glucose and beta D glucose they are anomers of each other. As I have told you, alpha and beta are nothing but these are names for the anomers. Now reaction of glucose. When you will call alpha, you understand it. Whenever OH is down, up, you will say in that down, and it will be alpha. If it is up, beta. Reaction due to aldehyde. Now reaction due to aldehyde आप कैसे कर सकते हो? अब देखो, ये सब कुछ reactions हैं. Basically to find out that what is the structure of glucose, we had to go through some of the reactions. With those chemical reaction only, we predicted what is the structure of glucose. In NCERT, it is very beautifully explained. Please go through it. We will solve more and more number of questions better. And I will just show you the few things directly. I will be showing it. Now, glucose here is there when it reacts with red phosphorus. Because I have told you red phosphorus reduces every carbon oxygen bond. It doesn't matter that which kind of it is. Either it is C double bond O, C single bond O, doesn't matter. It just reduces everything. So if it reduces, what will happen? Here red phosphorus and HI will make it convert into N-hexane. If you talk about CH3COCl, if it reacts with CH3COCl, how first of all it looks like? देखो, C double bond O, H, OH, H, OH, H, OH, H, OH, H, and C, H, H, O, H. Can you see CO bond? How many of the CO carbon oxygen bonds are there? 1, 2, 3, 4, 5, 6. Now all of these CO bonds will be reduced. तो रेड फॉस्फोरस एंड HI, इसे हम लोग प्यार से और क्या बोलते हैं? सर्वसाफी. ये सबको साफ कर देता है, सबको साफ कर देगा. एवरी CO bond will be, so here it converts it into n hexane, so all the CO bond will be broken. और कितने कार्बन, 1 2 3 4 5 6, so it will be n hexane. CH3COCl, CH3COCl will be, देखो इसको ऐसे अटैक करा दो, CH3CO प्लस और Cl माइनस में ऐसे ब्रेक करके, और फिर क्या करा दो, जस्ट मिनट, CH3CO, हाँ ठीक है, प्लस माइनस ब्रेक करके इसको क्या करा दो, H प्लस में O प्लस O माइनस H प्लस से ब्रेक करा, से रिएक्ट करा करके COCH3 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 से अटैच करा दो. व्हाट यू विल गेट अल्टीमेटली, पेंटा acetate. That is, how many of the OH group are there? So there are 5 groups are there. So how many of the acetates will be formed? I hope you all can see that, penta acetates. NH2OH, how NH2OH reacts with? I hope you all can remember कि whenever we have studied, when we were studying about aldehyde ketones, or we saw question on that basis, H2NZ. If let's say was there, what was happening, H2O को बाहर करो and it will convert into NZ. अब यहाँ पे N कुछ नहीं, ये क्या है, NOH. ये क्या इसमें कन्वर्ट हो जाता है, ऑक्साइम में, इट कन्वर्ट्स इनटू ऑक्साइम, तो इसलिए इसे क्या बोलते हो, ऑक्साइम. HCN से रिएक्ट करेगा, तो HCN किससे रिएक्ट करता है, आई होप अगेन यू विल सी H प्लस CN माइनस इज देयर, तो व्हाट इट विल ट्राई टू डू, इट विल रिएक्ट विथ अगेन CO. कैसे रिएक्ट करता है, कैन यू सी, इज देयर एनी कार्बोनाइल, यस, कार्बोनाइल पार्ट में we know that, how do you do that, plus if there's, let's say CN is there, so it basically reacts with and converts into cyanohydrin, it converts into cyanohydrin, so glucose cyanohydrin will be formed. आगे बढ़ो, HiO4, if you react it with HiO4, it will convert into HCHO and HCOOH, so basically it will oxidize all of these carbon and break all the bonds, so HiO4 converting into HCHO and HCOH. Schiff's reagent or NaHSO3 or all these things, please remember, because they are very large, so they cannot attack so easily. Why so? Because it is very much hindered, so in such case it cannot reduce through it, so there will be no reaction. So these reactions indicate that the glucose has 6 carbon straight chain with 1 CHO group and 5 OH groups. So these reactions were used to find out the structure of the glucose, but the structure of glucose was not only straight chain, but it was also found in the cyclic structure, though I have already told you, but it was scientifically it was proven, right? So what were the few questions कि अब ऐसा क्या हुआ, क्यों वो reaction नहीं किया, that's why people tried to find out, and they found out that this is hindered. Why it is hindered? Because of this formation of the cycle, right, और ring. चलो अब देखो, अब वही अगर ग्लूकोज का रिएक्शन तुम ब्रोमीन और H2O से रिएक्ट कराओ, तो इट इज व्हाट, इट इज अ माइल्ड ऑक्सीडाइजिंग एजेंट, इट इज नॉट अ स्ट्रॉन्ग ऑक्सीडाइजिंग एजेंट. अगर माइल्ड ऑक्सीडाइजिंग एजेंट है तो ये क्या करेगा, इट विल ओनली कन्वर्ट वट एवर, कंसिडर करो एल्डिहाइड पार्ट है उसको किस में कन्वर्ट कर देगा, एसिड में. इट कैन नॉट कन्वर्ट एल्कोहल इनटू एसिड, ओनली एल्डिहाइड टू एसिड, तो हियर ओनली वन चेंज, तो इसे क्या बोलते हो, ग्लूकोनिक एसिड. बट इफ इट रिएक्ट्स विद टॉलेंस रिएजेंट, I hope ammonical AgNO3 is there, what will be formation, because Ag plus converts into Ag and ammonium ion जो part है वो यहाँ पर converted with reacts with the CO part, it will be formation Fehling's से sodium का salt बन जाता है. HNO3, remember HNO3 being a strong oxidizing agent, ये क्या कर देगा, it will oxidize whatever is the 1 degree alcohol is there, as well as whatever is the ketone or aldehyde is there. Is there any aldehyde, yes or no? Yes, of course we know that aldehyde top पर है तो वो COH में कन्वर्ट हो गया. 1 डिग्री अल्कोहल है क्या, हाँ है, उसको भी COH में कन्वर्ट कर दिया, इसे क्या बोलते हो, सैकरिक एसिड. प्लीज कीप इन माइंड HNO3 वर्सेस Br2 and H2O. क्वेश्चंस इज आस्क्ड, what inform in the presence of Br2 H2O, gluconic acid, in presence of HNO3 saccharic acid. Very important question comes. आगे बढ़ो. Now which of the following sets of monosaccharides form sucrose? सुक्रोज क्या बनाता है भाई, देखो सुक्रोज इज मेड अप ऑफ फ्रक्टोज एंड ग्लूकोज, बट इन विच फॉर्म, प्लीज रिमेंबर, अल्फा डी ग्लूकोपायरानोज एंड बीटा डी फ्रक्टोफ्यूरानोज, नॉट पायरानोज, बीटा अल्फा डी ग्लूकोपायरानोज एंड बीटा डी फ्रक्टोफ्यूरानोज. हाउ डू यू सी दैट, चलो मैं तुमको दिखा देता हूँ. पहले जरा सोचो कि अल्फा डी ग्लूकोपायरानोज कैसा दिखेगा. अल्फा मतलब OH इज अप और डाउन, आई होप यू ऑल कैन सी दैट, OH विल बी डाउन, ठीक है, HOH. यहां पर भी नेक्स्ट फर्स्ट कार्बन, सेकेंड कार्बन, द हाइड्रोजन वाज राइट और लेफ्ट, दिस, देन OH H, then OH H, then CH2 OH H. This is what, this is beta D, sorry, alpha D glucopyranose. What about beta हाँ, can you see here? Now we are going to talk about next, that is beta D fructofuranose. Now it is not pyranose but it is furanose. How it will be looking like? OH, and here it will be CH2 OH, here it will be H1 1 2 3 3, here OH up down, OH down up, CH2 OH H. Can you see? Now here we have OH and H. क्या कर दो इसमें से? वाटर को रिमूव करा दो, और वाटर को रिमूव कराने के बाद यहां से क्या बना दो? ग्लाइकोसिडिक लिंकेज. दिस विल बी ग्लाइकोसिडिक लिंकेज, ग्लाइकोसिडिक लिंकेज फॉर्मेशन. और ग्लाइकोसिडिक लिंकेज फॉर्मेशन से बन क्या जाएगा? सुक्रोज. क्या बन जाएगा? दिस विल बी फॉर्मेशन ऑफ सुक्रोज. तो अल्फा डी ग्लूकोपाइरानेज एंड beta D fructofuranose gives you the, I hope you all can see that here alpha OH is down, alpha OH is up, so it is what? Beta D fructofuranose. So how many number of carbons are there? 5. Not 5 carbon, but number of members in the ring is how much? There are 5. So therefore it is furanose, not pyranose. Okay, keep in mind. Next आगे बढ़ेंगे. The number of atoms in the cyclic structure of D fructose is. Now D fructose मतलब D fructose के cyclic structure में कितने member हो गए जो कि जरा आप यहीं से चेक कर लो. Can you see that? Is this carbon chiral or not? Yes, of course this is chiral. Is this chiral carbon? Yes or no? Is this chiral carbon? Yes or no? Is this chiral carbon? Yes. How many are there? 4. So always remember that I am writing, it is going to be atom in the cyclic structure of D fructose. The number of atom in the cyclic structure of number of atom in the number of atom only. So it is what? It is 5. We have already seen. एक बात याद रखना, chiral carbon in straight chain of glucose. Glucose is going to be 4. Fructose में. So these points just keep in mind. Directly if the question comes, put the answer. And chiral carbon in a ring form of glucose is how much? It is going to be 1 more. 4 plus 1 is 5. While for fructose it will be how much? 1 more. Fructose is how much? It is going to be 4. Why? Because the carbon which was here, there in ketone and aldehyde were there, which were or we can say achiral. Now they also converted into chiral carbon. Why? Because they are now taking part in the glycoside, what you say, that in the hemiacetal formation. So glucose now in chiral carbon in ring form of glucose is 5, fructose is 4. Keep in mind, question may directly come from here. आगे बढ़ो. In fructose the possible optical isomers are how many? Now सोचो अब कितने होते हैं भाई. अगर आप सोचो कि कितने होते हैं optical isomers, it is asking for fructose में if nothing is given तो open chain. Which chain? Open chain. अब open chain में जरा सोचो कि how many carbons are there? There are 3 carbons. So 2 to the power 3 is how much? 8 is the correct option. आगे बढ़ो. Now classification of amino acids on the basis of, now we are going to talk about amino acids. I hope you all know that what are amino acids. Amino acids are the something due to which, or they are the smallest units of the protein. You know, proteins को form करने में amino acids are formed. There are 2 types of amino acids on the basis of requirement, that is essential, non-essential, but last one is semi-essential. Semi-essentials are only few that you have to keep in mind, like histidine and arginine. Histidine and arginine. We'll talk about others also. Okay. Next, on the basis of occurrence, there is natural and synthetic. Chemical nature is neutral, acidic, or basic. Neutral when one COH and one NH2 is there. Acidic when more than one COH and one NH2 is there. Basic when more NH2 and less COH is there, means more than one NH2 and one COH is there. Then this will be basic. I hope you all understand it. Based on relative position of COH and NH2, I hope you all know that. Here, what is the first of all general formula you should keep in mind? What is general form? General form is something like that. देखो मैं जो बना रहा हूँ, very important. 1 carbon make it attach with NH2 COOH 1 H and 1 R. This R can be anything. R can be anything. So one COH is there, one NH2 is there, one hydrogen, one R is there. So what we can say, what is the main functional group? One main functional group is going to be COH. If this is main functional group, this will be alpha carbon. तो अल्फा कार्बन पे अगर NH2 होगा तो इसे क्या बोलोगे? अल्फा अमीनो एसिड. अगर वही NH2 अगर बीटा कार्बन पे लग गया तो बीटा अमीनो एसिड. आई होप यू अंडरस्टैंड इट. अगर गामा कार्बन पे लग गया तो गामा अमीनो एसिड. नॉट अ बिग डील. आगे बढ़ जाओ. वही चीज यहाँ पे दे रखा है. So can you see it is alpha carbon, it is beta carbon, it is gamma carbon, it is what? Delta carbon. On that basis, it is converting into alpha, beta, gamma, delta amino acid. Okay. Now, classification based on requirement. What is essential and what is non-essential? Essential is something that you have to take from outside source. It means body cannot synthesize it. And what is non-essential? That it is not needed to take in from outside because our body can synthesize it. And what about the semi-essentials? देखो semi-essential ऐसे हैं that body also forms, but it may be needing it when there is something need, like you know, like you can see if there is periods of the girls are there, or if there is, let's say, there are many cases like, you know, like if there is a, if kids are growing, so these growing stages also are there. So that time whatever is the amino acids are there which are needed to be taken from outside, it is called semi essential, though they are formed in our body. Okay, chalo, ये सारी चीजें I hope you all understand it. Now classification based on the chemical nature, like R, CH, COH, NH2. These are the things that you have to understand. This is the general form of the, you can see, amino acids. Now if R contains COH, please remember, if this R contains COOH, then one COH is already there, one more COH will be there, so it will be acidic amino acid. If this R contains NH2, so one NH2 is already there, one more NH2, then it will become basic, because NH2 is what? Basic. Why is it so? Because nitrogen has the lone pair that it can give to any H ion. I hope you all understand it. If R neither contains COH nor NH2, so there will be one COH and one NH2, means one acidic part, one basic part. So it will be neutral amino acid. Is there any doubt? I hope there is no. Now acidic amino acids, what are the acidic and basic amino acids? You have to keep in mind. So if you talk about acidic amino acids, there are two, that is aspartic and glutamic acid. Aspartic and glutamic acid, what are the formula? यार aspartic में सिर्फ बीच में एक CH2 लगा देना, means yes NH2, ये तो सब में सेम है, यहाँ पे CH2 COH, यहाँ पर क्या कर देना, 2 times CH2 लगा दो, so 2 times CH2 ऐड कर दो, same चीज. So this will be what? I hope you understand. So therefore it is what? What is isoelectric point? I will tell you. What is isoelectric point? I will tell you after some times. Next, चलो आगे बढ़ेंगे. Now lysine, arginine and histidine. These are lysine, arginine and histidine. These are what? These are basic amino acid. Basic amino acids means there are going to be more NH2 and less COH. Means in the carbon chain there is NH2 extra. Yes or no? Can you see? There is extra NH2. Can you see? So therefore, so wherever there is going to be one basic nitrogen is going to be there. So that is what it is basic. So what are their PI, that is isoelectric points? Can you see? It is more than 7. So that's why they are basic. If it is less than 7, it will be not always, but acidic generally. Now always remember, this is something that you can keep in mind, or you can take a screenshot, or you can see this video. Very important. Here red means it is aliphatic. This green color is aromatic. Aromatics very important. Sometimes you have to keep in mind what are these: phenylalanine, tryptophan and tyrosine. These three are aromatic. Question comes from here. Next, acidic, basic. Acidic के लिए देख लो aspartic acid and glutamic acid. Basic, you can see that arginine is there, histidine is there and lysine is there. Yes or no? Next आगे बढ़ो. Sulfur containing. Sulfur containing में cysteine and methionine. Cysteine का भाई cysteine भी होता है, though it is not considered because cysteine का ही अगर तुमने इसे क्या कर दिया, dimerize कर दिया, you will get cysteine, right? So this is also sulfur containing. Amidic, so you can see that which of them contains amide, that is CONH2, that is asparagine and glutamine. important है. नॉन एसेंशियल एंड एसेंशियल, देखो कहीं से क्वेश्चन आया ना आया, एक जगह से क्वेश्चन बहुत आता है, इज इट एसेंशियल और नॉन एसेंशियल. सो आई विल बी टेलिंग यू हाउ टू डू और व्हाट इज द ट्रिक फॉर दैट. ओके, चलो आगे बढ़ेंगे. सो बिफोर दैट एक ट्रिक याद रख लो. ट्रिक फॉर यू नो एसेंशियल्स अमीनो एसिड. एसेंशियल amino acids. What are the essential amino acids? Please keep in mind. कैसे इसे याद रखोगे? समझो, you know, you are in the college, there are TT, table tennis are there. So wherever the table tennis is there, it is called TT hall, right? So अगर मान लो टीटी हॉल में कोई वीआईपी आ जाए तो क्या बोलोगे? टीटी हॉल में वीआईपी. कैसे याद रखोगे? टीटी हॉल में वीआईपी. टीटी हॉल HALL में ME, that is this E is not, you know, ME, may VIP. And please keep these points in name, these names in your head, so rest of them will become non-essential. So TT के लिए क्या रखना है भाई? tryptophan, threonine, H for histidine, A for arginine, L for lysine, L for leucine, Me for methionine, V for valine, I for isoleucine, P for phenylalanine. Can you see? देखो अभी अगर ये तुम्हें याद हो गया तो फिर remaining all of them will be non-essentials. So TT hall में VIP, T for tryptophan, threonine, H histidine, and A for arginine. Plus please remember that arginine and histidine are sometimes not considered as the essential because they are semi-essentials. Sometimes they are also kept in non-essentials. But please remember, if it is there, so please you can keep this as the essential in the option if it is given. आगे बढ़ो. Now, so next is isoelectric points of amino acids. देखो हमेशा कुछ बातें याद रखो. Isoelectric point क्या होता है? पहले तो आप इससे पहले ये समझो कि if let's say there is any isoelectric point, is something constant is there, some constant is there at some given temperature. If you are giving less pH than isoelectric point, so it will be acidic nature for or acidic environment for that amino acid. And if you are giving more, you know, more pH than that isoelectric point, so it will be basic environment for that amino acid. अब जरा समझो, एक अमीनो एसिड में आपका COH भी है और NH2 भी है. NH2 is basic and COH is what? Acidic. If acidic part is there, so if base is there, so it will leave its HN. Yes. So always remember. देखो जब भी आप PI that is PI is there. PI means isoelectric point. At isoelectric point, any of the amino acid we can see that amino acid will remain as the Zwitter ion. How it will remain? It will remain as Zwitter ion. It will remain as Zwitter ion. What is the specialty about Zwitter ion? It has positive and negative both the poles at the same time. Positive and negative both the poles at same time. When it will be a isoelectric point. Can you see? NH2 and NH2 has converted in NH3 plus and CO minus has converted in COH has converted in CO minus. So basically keep in mind it is what? Isoelectric point. अब यहां से क्या होगा? अब समझो यहां से तुमने इसे base दिया. If you give it base, so in the base what will be there? Can you see? There will be OH minus, or we can say that HIN will be less. If HIN will be less, in such case what will be there? So in the base this HIN will be taken. And as this HIN from this acid will be taken, it will convert to CO minus and NH2. Can you see? And what will be happening if you give it HIN? Let's say acidic environment if you give. So in such case, what will happen? Because NH2 is basic, so it will attract H plus and it will convert into NH3 plus. So please keep in mind, अगर आपने if the pH is less than PI, तो क्या हो जाएगा? It will remain as the positive. It will remain as the positive. While if you give pH more than PI, it will remain as negative. So it will be cation here and it will be acting as anion here. But at the isoelectric point, it will remain as the Zwitter ion here. कोई दिक्कत? No दिक्कत. Please keep in mind. Okay. आगे बढ़ेंगे directly. Like examples. Can you see this example? How many of the COH is there? 1. How many of the NH2 is there? 1. So can we say it is what? Neutral. So please keep in mind, it will be given in this form NH3 plus and इसके लिए दे देगा pKa1. What is that? pKa for that it will be pKa1 and for that it will be pKa 2. How do you find out pi in such case? So pi, that is isoelectric point, it will be given as pka 1 plus pka 2 divided by 2. So I hope it is very clear, not a big deal, बहुत easy है, कर लोगे. अब तुम बोलोगे सर NH3 NH2 तो basic होता है, it should be what? It should be pka, it should be pkb. Why it is pka1 is given? You know that. You can find out pka into pkb equals to 14. Consider pkb भी दिया तो उसे convert कर लेना किसमें? pk, sorry मैंने multiply कर दिया. pka plus pkb is going to be equal to 14. What is that? pka for any. Consider it is base, so it will be conjugate acid. Consider it is acid, so it will be what? For conjugate base. I hope you all understand. So consider it is for acid. If it is acid, so this pkb for conjugate base. Understanding it? So if pka for acid, तो इसके conjugate base के लिए if it is pkb, तो pka plus pkb always will be 14. It will be equal to 14. It is something from ionic equilibrium. It is already known things that we have to know. तो चलो अगर इसके लिए pkb भी देता है तो you can find out pka by doing 14 minus pkb, you will get the pk. So pk1 plus pk2 you will get, pi will be divided by 2, you will get that. Can you see here it is acidic or all these examples are acidic or basic? I hope you all can see that COH is there, COH is there. More than one COH is there, so it will be acidic or basic. It will be acidic. अगर acidic है, इसके लिए मान लो क्या दे दिया है, इसके लिए है pKa1. इस NH2 के लिए क्या हो गया? NH3 बेसिकली NH3 plus pKa2. इसके लिए क्या हो गया? मान लो pKa3. So in such case, what will be the pi for that? Pi for that will be pKa1 plus pKa. Cable take acid, do not take for base. If two acids are there, take only for acid. pKa1 plus pKa3 divided by 2. Again I will say, consider it is for pKa1 and for this it is pKa2. Do not consider for NH2 because it is more acids are there na. So इसके लिए pi will be pKa1 plus pKa2 divided by 2. What about this? Can you see it is acidic or basic? देखो एक COH है. How many NH2 is there? 1 and 2. So can what we can say? Consider इसके लिए क्या है? pKa1. इसके लिए क्या है? pKa2. इसके लिए क्या है? pKa3. So pKa, now there are two bases. So do not consider acid. Only take base. So pKa2 and pKa3 for basic. It is what? It is basic. For basic you will take only NH2. So you will take pKa2 plus pKa3 divided by 2. क्या कोई भी दिक्कत है? I hope you all can find out KIP, that is isoelectric point, easily. Now an optically inactive amino acid is, देखो याद रखो सारे amino acids are optically active other than only one, that is, I hope you all know, glycine. That is, you can say the most, or you can say simplest. Here R becomes equal to H. So what becomes? C, NH2, COOH and one H, one more H. So this becomes achiral. I hope you all understand it. Next, a compound that can form zwitterion. So we all know that they are what? All the amino acids. So amino acids में कौन? क्या glycine amino acid है? All others are not amino acids. A non-essential amino acids among the following is, देखो टीटी हॉल में वीआईपी सोचो, टीटी हॉल में वीआईपी, लाइसिन है क्या? हाँ. वैलिन वीआईपी वी. ल्यूसिन है क्या? HALL. यस, लाइसिन ल्यूसिन है. एलानिन था क्या? एलानिन था क्या? नहीं. A was for arginine, it was not for alanine. So alanine is what? Non-essential. I hope you all can see the total number of basic groups in the following form of lysine. जरा सोचो जरा. Can you see that this is what? It is not acidic form, it is basic form. Can you see it has given already? HN has given, so it is basic. Can you see it is what? It is having lone pair, so it is also basic. ये क्या है? NH3 plus. अरे NH3 plus को basic मत बोल देना. It is acidic. Why so? because it is what? NH3 plus. It has H to give. It cannot take any H because nitrogen has already taken 4 or made 4 bonds. It cannot make the 5th bond anyways. It is what? Acidic. How many of the basic groups are there? 2. This. I hope आगे बढ़ो, समझ रहे हो? Which of the followings are acidic? Can you see? जहाँ पे COH ज्यादा हो, so there is no basis. यहाँ पे क्या है? दोनों सेम है. यहाँ पे NH2 is more basic. It is what? Basic. COH. Can you see? No. Can you see here? How many COH are there? 2. So it will be acidic. Option D is the correct option. आगे बढ़ो. Which of the following is a basic amino acid? We have already studied about arginine, histidine and lysine. Is there any arginine, histidine, lysine? Option C is the correct option. Next, the non-essential amino acids are, देखो मैं लिख दे रहा हूँ, lysine है, arginine है and histidine. So these are the answers. Next, the non-essential amino acids among the following. Non-essential, TT hall में VIP में से जो नहीं है, that will be non-essentials. Now TT hall में VIP, V valine, L lysine, leucine, alanine. No, alanine is not there. Lysine, leucine, they were in the TT hall में VIP. So alanine is not there. A stands there for arginine. आगे बढ़ो. Amino acids undergo internal acid-base reaction to form zwitter ion. आगे बढ़ो. Yes or no? क्या हो जाता है? R, देखो, R C N H2 C O O H H. It converts into, here can you see, minus plus. Can you see there is lone pair? So what will be there? Lone pair. इसको ले लेगा भाई. Can you see? तो ये कैसा हो जाएगा? RCNH3 plus COO minus H. Can we see? So it is what? Zwitter ion. So this was acting as the base and it was act as the acid. तो ये acid base reaction हो गया कि नहीं? Internal acid base reaction to convert into, yes, zwitter ion. Next, in a protein molecule, various amino acids are linked together by, अब हम प्रोटीन पे आ चुके हैं, so they are linked by peptide bonds. If let's say 2 amino acids are there, it will be called dipeptide. If 3 amino acids are there, it will be called tripeptide. If 5 amino acids are there, pentapeptide. If more than 10 amino acids are there, polypeptide. I hope you all understand it. What is peptide bond? जरा ध्यान से समझ लो. जैसे कि कोई R C C O O H है, N H2 है, H है, और साथ ही साथ क्या है? NH2 NH2 H2 NH NH NH2 and COOH H R डैश है. अब यहाँ पे क्या कर दो? दोनों को ऐड करने के लिए क्या कर दो? यहाँ से OH और H लेकर के यहाँ से क्या कर दो? माइनस वाटर रिमूव करो. R C NH2 H C O NH. Can you see what is there? CONH CONH C COOH H R dash. It is formed. So in such case, can you see this part? CONH. It is only called peptide bond or peptide linkage. Peptide linkage. Peptide linkage. Can we see? So आगे बढ़ो. चलो. Proteins are found to have 2 different types of secondary structure, which is alpha helix and beta pleated sheet structure. Alpha helix structure of protein is stabilized by, कैसे करते हैं भाई? Hydrogen bonding से. You have to read and through NCERT. These points are very well given there. Next, each polypeptide in protein has amino acid linked with each other in a specific sequence. This sequence of amino acid is said to be primary structure. Always remember, whenever you do the denaturation of the protein through heating or through any way, so what happens? Only the primary structure remains intact. All secondary, tertiary and quaternary structures get broken, okay, or denaturalized. When of which of the following statement is not correct? Now which is not correct? Think about it. Ovalbumin is a simple food reserve in egg white. Yes or no? Yes. Blood proteins thrombin and fibrinogen are involved in blood clotting. हाँ भाई, बिल्कुल सही बात है. I hope, क्योंकि यार तुम लोग तो यार बॉटनी zoology वाले हो. You have already studied about it. Blood clotting का whole mechanism. Now denaturation makes the protein more active. No, it makes it deactivated. not activated. Next, for this peptide bond, which statement is incorrect about peptide bond? जरा देखो, CN bond length in protein is longer than usual bond length of CN. देखो एक बात याद रखना, यहाँ पर जो CN bond है ना, there is going to be a kind of, can you see क्या हो रहा है यहाँ पे, conjugation, and due to conjugation what will happen? What will happen? अरे due to conjugation what will happen? This will be CO minus double bond NH. देखो अब अगर इसे बनाना चाहो, it should be something looking like that, NHCO CO, और इसके बीच में क्या आ चुका है, there is partial double bond character. Due to partial double bond character, due to partial double bond character, what we can say that bond length decreases, not increases, and bond strength increases. चलो आ जाओ, तो CN bond length in proteins is longer, no, shorter, no, shorter. Spectroscopic analysis shows that planar structure of CONH group. Yes or no? Yes, because this nitrogen is no more NH, it is not no more sp3, it is sp2. So sp2 means it is planar, not non-planar. CN bond length in protein is smaller than usual bond length of the CN bond. Correct है कि नहीं? बिल्कुल सही बात है. तो option C. So it is asking, अच्छा it is asking for incorrect. Okay, तो incorrect तो यही है, ठीक है, ये तो correct है, छोड़ दो भाई, आगे बढ़ो. Now nucleic acid क्या होते हैं? Nucleic acids are those, you know, those things inside our, you know, cells that just keeps our hereditary things to the next, you know, that just tells or talks about the heredity of, I hope you all know about it. I do not need to tell you because you know it better than me. आगे बढ़ेंगे, सीधे बातें करेंगे bases in the DNA, the chemistry of this part. Okay, so bases that is there in DNA and RNA. DNA में कौन कौन से होते हैं? I hope you know. DNA is nothing but deoxyribonucleic acid, while RNA for riboxynucleic acid. Now DNA में कौन कौन से bases होते हैं? Guanine, cytosine and thiamine. अरे एक मिनट, adenine, guanine, adenine, guanine, cytosine and thiamine. While in RNA there is adenine, guanine, cytosine and uracil. So here thymine is there but uracil is not there in DNA, while here in RNA thymine is not there but uracil is there. Please keep in mind, very important. क्या है इन सब का structure? It is important to keep in mind, please. It is there, you can take the screenshots, not a big deal, you can take that. ठीक है, should I? चलो आगे बढ़ेंगे. Next, the bases present in the, देखो इसके बारे में, even in इसके structure के बारे में there is a, you know, we have to study even better, but because of, you know, these things are there, keep in mind. Only right now I will tell you, please keep in mind, though I give you some of the what to say some of the tricks also, but if the question comes we'll see, otherwise okay, you can just remember that the base present in RNA but not found in DNA. DNA RNA में तो है पर DNA में नहीं है. Think about it, what we have seen that RNA में कौन कौन था? Adenine, guanine, cytosine and uracil, but here it was adenine, guanine, cytosine and thiamine. So there RNA में तो है पर DNA में नहीं है, that is uracil. आगे बढ़ो. The coupling between the bases unit of DNA is through hydrogen bonding. Yes or no? Yes. आगे बढ़ो. The correct statement of the regarding RNA and DNA respectively. The sugar component of RNA is arabinose. No. The sugar components of RNA is 2-deoxyribose. No, it is wrong. The sugar component of RNA is arabinose. No. The sugar component of RNA is ribose. Yes, ribonucleic acid, that's why we give. And sugar component of DNA is 2-deoxyribose. Very important. Yes. Now in DNA the complementary bases are, which are the complementary bases? देखो जैसे वो जिनसे भी attach होते हैं, they are called complementary bases. Now adenine and, you have to remember, adenine and thiamine, while cytosine and guanine are attached like that with the 2 and 3 hydrogen bonds, you can say adenine and thiamine and guanine and cytosine in DNA. आगे बढ़ो. Now if we will talk about the vitamins. What are the vitamins? About the vitamins, I hope you all know about what are the vitamins, right? So vitamins are accessory food factors required in the diet. They are classified as the fat soluble, that is A, D, E, K, and water soluble only B and C, while the water solubles are B and C. Deficiency of vitamins leads to many diseases. These diseases you have to keep in mind. Their chemical names you have to keep in, or you can say their scientific name you have to keep in mind, like vitamin K, vitamin E, vitamin D, vitamin A. Vitamin A is retinol, vitamin D is calciferol, it is called calciferol. What about E? It is tocopherol, tocopherol. What about this? This is phylloclovin, phyloquinone, we can say phyloquinone is vitamin K. What does this B? B has the name, the whole, B1, B2, B2, the whole spectrum of the B vitamins are there, like B1 is for thiamine, B2 is for riboflavin, and and B3 for niacin, B5 for pantothenic, B6 for pyridoxine, B7 for biotin, B9 for folate, B12 for cyanocobalamin, basically carbon, C for ascorbic acid. So these are water soluble, these are fat soluble. Any doubt? आगे बढ़ो. Next, these names should be please remembered. Question may come from here. Now, what are the sources? It is written. What are the deficiency diseases? It is also written. You have to keep in mind. आगे बढ़ेंगे. These things again, same things are there. Keep in mind. Not a big deal. Next, vitamin B12 contains cobalt, cyanocobalamin, because it has cobalt. Fe+2 कहाँ पे होता है? Hemoglobin and myoglobin में. ठीक है? आगे बढ़ो. Which of the following vitamins is water soluble? Water soluble कौन है? B and C. Is there B? Yes. Scurvy is caused due to the deficiency of which vitamin? Option C. That is vitamin C. So that was all for today class. So it was the last lecture for this series. That was rapid revision session. I would just suggest few things before going or before taking a leave. A few things please keep in mind, because now just not more days, just 6 days are there. More there will be, you will be there in the examination hall. Very important, next 4 to 5 days are there. What you have to keep in mind and what you have to do before the examination, during the examination, and right now in these 5 days. Please keep in mind few things. First of all, do not study now for next 5 days. Please do not study. Don't study anything new. Anything new. Second thing, important है भाई, ऐसा भी नहीं होना चाहिए यार कुछ छूट गया है, अभी छूट गया है, अरे बोला पूरा पार्ट छूट गया है. प्लीज जो छूट गया उसे रहने दो. Please remember that no student is going to be 100% prepared here. Of course, there will be few, but okay. चलो वो 720 लेकर के आएंगे. Okay, you have to at least go for targeting 600 to 650, and for that it is very much important कि last time में do not study anything new. First thing. Second thing, कि whenever you are thinking of कि यार अब क्या होगा, I have not gone through it. No, don't. Please come to always to one point. क्या कुछ नया पढ़ लूं, क्या कोई नया टेस्ट दे दूं? No. Don't take any new tests. कोई नया टेस्ट अब मत देना. Only revise all the tests and योर मिस्टेक्स. उसमें जो भी आपके मिस्टेक रहे उनको रिवाइज करो और दोबारा ना हो इस बात को मेक श्योर करो, इंश्योर करो. नेक्स्ट, व्हाट आर द नेक्स्ट मेन पॉइंट्स? टेक केयर ऑफ योर हेल्थ. ठीक है, तो प्लीज टेक केयर ऑफ योर हेल्थ. टेक केयर ऑफ हेल्थ. इन टेकिंग केयर ऑफ योर हेल्थ, मोस्ट इंपॉर्टेंट थिंग इज कि ड्रिंक वाटर. भाई अच्छे से ड्रिंक वाटर तो करो, वाटर इनटेक अच्छा खासा रखो. नाउ डोंट ईट ऑयली फूड उतने दिन के लिए. फॉर द नेक्स्ट 5-6 days do not eat any oily food. And about the mental health, what about the mental health? देखो कुछ बातें मैं बोलना चाहता हूँ for mental health and keeping yourself positive enough. याद रखो, if you feel कि I have not 100% prepared, nobody is prepared, please believe it and make yourself keep on talking to you. It's okay, I'm fully prepared and I will be doing my best. Whatever I'm prepared with, I will not let anything be negative. ऐसा नहीं होना चाहिए कि you are prepared for let's say 600 but due to the negative marking you came to 550 540. It will be very it will be pricking thing. So please take care about it. उसके लिए जरूरी क्या है? Your mindset should be positive rather than trying to focus on something that you have not done. Try to focus on things that you have already done. So here first of all I will tell you, revise only that too strongest and moderate topics at last time में अब और कुछ भी नया नहीं करना है. Next one more few thing कि mental के लिए क्या करना है? Talk positive to yourself. Talk positive affirmations. See, the number of days are very less, though I tell this thing to do to every student back from starting only, but still what you have to do, still there are a lot many days. You can keep yourself, अपने दिमाग को उस zone में लाओ. कैसे? Do meditation or do whatever you want to do and think like कि what will be the day, one day before the examination, how it will be. I will be relaxed, I will be fully in my control, I will not be panicking, I will not be thinking of something that I have not prepared. I am very much prepared and whatever I am prepared, I am going to do best out of it. Think about it. Then think about कि how do I go to the examination hall. When you are going in the examination hall, think about it. कि when I go to the examination hall, I will be reaching there on time. There will be no rush. I will be leaving my home at this time. I will be reaching by this time and everything will be very fine. I will go, I'll relax there by going in the classroom. When the paper comes, अब जरा सोचो, पेपर से पहले सोना मत. Do not sleep before the paper. What happens? This may decrease your energy level. Never ever try to do this mistake. Second thing, during the examination, there may be two possibilities. One, the paper may be difficult or it may be easy. If it is easy, remember you have to think of getting more and more marks. How? By being a bit faster but at the same time accurate. फास्ट होने में गलती से भी ये गलती मत करना कि if there is one question you have hurried up, you have just ticked the answer, you have not cross checked it. Please do not do that mistake. Check it. कई बार ये लगता है बहुत इजी क्वेश्चन होता है ना, एक बार पक्का देख लो कहीं करेक्ट तो क्वेश्चन तो ऐसा नहीं था. I am taking correct option but it is written incorrect. I am doing decreasing order but here it is increasing order. I am doing yes but it was giving no. देख लेना प्लीज, बहुत इंपॉर्टेंट है ये सारी बातें during the examination करना. Then during the examination think more things. ये सारी चीजें अभी से ही सोचो, ऐसा नहीं कि वहां पर जाके होंगी. अपने दिमाग को उसमें डालो. Just give those affirmations to yourself when I'm in the examination. If it is easy, try to gain more and more marks. चलो मान लो कि टफ पेपर हो गया. Let's say 3, 4, 5 question in straight line you could not solve. मतलब एक बार एक पूरा स्ट्रिक बन गया, 2, 3 question you could not solve in chemistry or physics. It will give a lot of pressure to you, but do not get pressurized. Please remember, if it is tough for you, it is tough for everyone. So if the paper is tough for you, it is tough for everyone. A very very important point that students forget and they think अरे यार मुझे तो 700 ही लाना है. अरे यार जब टफ हो गया पेपर तो सबके लिए टफ हो जाएगा ना, तो 700 लाना इज अ टफ थिंग. एनीवेज वो जो एक्स्ट्रा 5 क्वेश्चन थे वो हट चुके हैं, तो इट विल बी वेरी टफ. तो प्लीज कीप इन माइंड कि अब पेपर बिकम्स टफ, more focus should be on the accuracy, not on the more number of questions. Keep in mind, when I say a paper is tough not because of chemistry and physics is tough. When you see कि botany is also becoming very hard, right? Then you say that paper is tough. And so these are the points that you have to keep in mind. चलो कई बार होगा कि 1, 2, 3 3 question you could not solve at once. तो क्या करो ना, now break the streak. Rather than going from that order, just change the order. Or let's say you are going from 1, 2, 3, 4, 5. No, start from 45, 44 like that. Okay. Or you may think just completely change that. Completely change the subject. You can go like that also. Whatever you feel like, please do like that. And whenever you are solving the paper, because it is paper pen paper question paper, so please, it is done in 3 steps. First step, see all the easy questions. Easy question को पहले कर लो. Within 15 minutes, you will easily solve 20 to 25 questions. इट कैन बी डन. चलो उतना नहीं सही, 20 हो जाएगा, 20 मिनट्स में इतना हो जाएगा. रिमेनिंग क्वेश्चन शुड बी लेफ्ट फॉर मॉडरेट. मॉडरेट आर दोज जिसको आपको पता है कि यू विल हैव टू सॉल्व, यू विल हैव टू गो थ्रू 2-3 प्रोसेस इन फिजिक्स एंड केमिस्ट्री स्पेशली. सो keep it for the second step of the solving, and third step will be at last. Means वो question जो बहुत tough है, that you are not getting any idea कि how to solve it, तो ये सबसे last में रखना. Okay, so first easy, then moderate, and at last you will solve the tough questions. So please do things. So अभी से ही positive affirmations सोचो, करो, and meditate and talk to your parents. Very important. Talk to your parents, or you can say whoever is there with you, who is someone who gives you positive vibes, not with someone who is giving you negative vibes. Okay, so these things keep in mind. So after all, it is your examination. You have to be strong enough to go there and rock it. So just few things I will tell you. Just think about that day also when you are visualizing. Visualize that also. Visualize something. Yeah, what if I just crack the NEET examination? What will be a beautiful day? What will be that day? Consider your name is there, you are qualified. Arey wow, your parents, you, how happy you would be. It will be a great feeling. You can't even imagine. Not just you will be feeling so happy on just above the seventh heaven. Means it is just above anything. And at the same time, those who are not, those who have, you have felt, yeah, this person has not thought beautifully for me. So think about it. If they will become so sad about you, oh my god, so think about that happiness also. Everything comes. So once you qualify for that, it will be a all game changer things. So please follow these things, follow these steps. And one more thing I was talking about, that visualize the exam day and day before examination and result day. यहां पर प्रेशर कभी मत ले लेना. यहां से do not get pressurized. Don't get pressurized by the content you have not prepared. जितने भी PYQ एंड अलग अलग क्वेश्चन पेपर से आपने इंपॉर्टेंट क्वेश्चंस दैट यू हैव encircled, that questions, some important questions are there that you encircle, please go through them, solve them, and that will be all for your preparation. And after that, you just go in the examination, and rather than thinking of what if I do not do, or what if the positive things are not there, do not. Go and enjoy the day. Go and enjoy those 3 hours, and just come like you have won. Whatever you had, at least you should have that feeling कि मुझे जितना आता था, I have done it all, and I have done it positively. I have not made any negative markings. Negative marking ना करने के लिए मैंने सीधा बात बताया. Please 5 seconds extra दे दो, 10 seconds extra दे दो, but do not give -5 marking. सोचो, जो आप 5-10 seconds extra नहीं देते हो, you just lose -5. You should just lose 5 marks. It's a very big deal. Rather than losing 5 marks, lose 5 seconds. 5-10 seconds extra दे दो एक क्वेश्चन पे, but please do not lose 5 marks. Negative marking कम करो, and let me tell you, this will give you immense confidence. और उसके बाद जो आपके साथ होगा, the whole paper will be a good positive outcome. Okay, so that is all from my side. All the very best, all the very best, my fellow students. I have really liked this journey with you. Thanks for joining in all these ways.